Welcome back. We are continuing our discussion on neurophotonics, and today I have another topic for you, and that is neuroimaging with light sheet microscopy. It is also sometimes is used as LSM, but LSM has other connotation, especially in life science. Laser scanning microscopy is also called LSM. So, this is usually LISM, but overall we are going to discuss light sheet microscopy, not laser scanning microscopy. So, understand this, that there are several challenges when you are trying to understand the working of a neurons, or when you are trying to image neurons specifically in a live organism without taking the brain out and without any sort of dissection. So, achieving a balance between high spatial and temporal resolution in neuroimaging is very challenging. Neuroimaging often deals with weak signal amidst background noise, and there is a huge amount of inter-individual variability in brain structure. Interpreting neuroimaging data requires expertise in both neuroscience as well as imaging analysis, and therefore misinterpretation is very common. So, in 18th century, early 19th century, the gentleman Golgi, what he did was he used to dissect animals, the brains of the animals, try to dissect neurons, those nerves, not neurons, nerves of the animals, cover them with some sort of silver salts, and then put the entire brain structure, entire nervous system under the microscope, shine light and try to see the staining that has happened by silver salts, right, from which he came up with, Golgi especially came up with all those beautiful axons, dendrites definition, then synaptic cleft. Golgi is the same person from which Golgi apparatus or Golgi bodies are named. It is the same scientist. So, Cayal, Spanish scientist, also work similarly, where they used to put silver salt, staining the brain with silver salt, hopefully the brain of animal which is no longer alive, and then try to image it. But all of those functions were difficult, caused errors, gave weak signals, and it is very subjective regarding what you are looking at, and the small change in ions depending on the type of neural circuitry going on, what sort of electrical impulses, action potential has been given. So, it causes huge amount of challenges that needs to be overcome. Light sheet microscopy, it was said, that could overcome several of these problems. Now, light sheet microscopy is not at all recent. In 1902, German scientist, frankly speaking, I hope they are German and I cannot pronounce their name. You try if you can pronounce their name. This looks like Polish to me, but this looks like German to me. So, I am just guessing. These two gentlemen came up with the term ultra microscope. So, what they did, they saturated certain areas, certain samples with metal nanoparticles, metallic particles, and they illuminated the sample. They illuminated the sample from say top. They shine light on the sample from top, but the detection, the measurement was from a orthogonal 90-degree angle. So, if you are shining light from the top, you are measuring the sample scattering, the light scattered from the side, 90-degree difference. This is input, this is output, or if this is illumination, you are measuring it from up or measuring it from bottom. Usually what we have in a reflected microscope, light comes, falls and returns back. So, the entry and the output path is exactly the same. It measures like you capture the same light and then you try to analyze it, measure it at normal reflection microscope. In transmission microscope, light falls, into a sample, it penetrates through it, it transmits through it, and you have your detector below and you are trying to measure it. Here it is 180 degree, light is falling and you are measuring it from an orthogonal position. And they said that this prevents, now it seems very common sense to you why we do not do it, but as you see that the cost becomes very high. The light has to fall from here. If you measure it from an angle or measure it at a 90 degree or any other angle per se, you are eliminating any other noise, any other scattering that is caused by the input light. Simple. So, it was based on an idea long time ago and they called it ultra microscope. This is that ultra microscope image, and they claim that they help scientists to see really small things clearly like cells or particles. So, basically this is what is happening. This is the sample box where you have your particular sample and it is illuminated from this side, it is illuminated from this side, it is illuminated from this side, and at a 90 degree you are doing the detection. It is illuminated from this side. It does not have to be like this, it can be like this, but it can also be like this. X and Y are at 90 degree to one another, X and Y coordinates. X and Y coordinates are 90 degree to one another, X and Z coordinates are also 90 degree to one another, yes. Yeah. So, this is the principle of light sheet microscopy. It utilizes a specific optical setup to achieve high resolution imaging. It is imaging technique that involves using a thin sheet of light to selectively excite fluorophores. This focuses excitation minimized photo bleaching and photo damage, and the emitted fluorescence from the illuminated plane is then collected by detection objective. So, again let us try to understand what is happening. You have understood the old light sheet microscope technique. It illuminated from one side and capture the information. The detection is from a 90 degree orthogonal angle. You can shine the sample could be, the light could be shined here and you can detect it from there. What presently people are doing using some sort of cylindrical lenses, which I will show you, the illuminating light is not just one single dot, usually it is a point. Yeah, when you are illuminating a sample, it is usually a small spot, usually circular, that falls onto an area, it gets reflect, transmitted, etc. Using cylindrical lens, they are trying to make it a squarish or rectangular, like a sheet of paper. A light falls, a laser light falls like in a sheet, not just one point, not just a sphere, not just a circle, it is a sheet. That is the sheet of light sheet microscopy. The laser which is coming like that, this is a spot, this is a spot. Instead of a spot, you use lenses so that the laser light makes a rectangular, a sheet like structure that almost like slicing through the sample. That's like slicing through the sample. As it slices through the samples, it illuminates large number of area. Not just pixel by pixel, You are not trying to scan individual areas, individual pixels like you have done before. You are scanning an entire area using a particular wavelength or set of wavelengths. As the light is slowly moved in this particular direction, you are measuring the detector at an orthogonal angle, at a 90 degree angle in the same manner. But if the light is falling in this direction, your detection is in this direction. If the light is falling in this direction, your detection is in this direction. And it helps prevent several problems. This focused excitation minimized photo bleaching, photo damage to the region outside the illuminated plane, and it provides you a 3D imaging and a very high speed data rate. You are not imaging pixel by pixel, you are simply scanning plane by plane. Instead of using pixel, you are sending a plane, a sample is getting dissected by light, not literal dissection. You simply are illuminating it plane by plane and measuring it from side to side. So, this is something that is happening. You have a sample, you have an excitation laser system. You have a cylindrical lens. The cylindrical lens produces the point source of light into this kind of a light plane, a beam of light that is squarish or rectangulish, and it illuminates the area. It almost slices, optically slicing the sample into like planes rather than points, and you have this objective that tries to see the light that is being scattered when a plane by plane, the light is moved or the sample is moved, and the scattered light is collected by the cylindrical lens, put into camera, CMOS camera, etc., and you are simply measuring it, analyzing it using a computer. So, basically in light sheet microscope we are producing plane, a sheet of light. The sheet of light is made using cylindrical lens. Cylindrical lens can convert a point source into this kind of rectangular source. The sample is slowly passed through the sheet of light. Different planes are sliced, different planes of the samples are illuminated, and whatever planes are illuminated, they scattered some amount of light. The scattered light are connected orthogonally at 90 degree. This is the direction of the plane of the excitement light. This is the direction of the plane. So, if this is z direction, this is I do not know y direction or x direction, whatever, and this is collecting it. So, illumination system is often a laser to generate the thin sheet of illumination. The light is usually directed through optical element like lenses, mirrors, and the beam shaping device to achieve desired thickness and shape of the light sheet. Sample holder is mounted using a precise positioning. Detection optics is some sort of an objective lens. The lens collects an emitted fluorescence, and for 3D imaging on multiple acquisition, light sheet fluorescence microscope, you have obviously used some sort of a fluorophore here to often include a sample scanning or mounting stage. The advantage here, understand, is as this sample, as this sphere is illuminated plane by plane and collected information, the scattered information is collected, you are able to get a 3-dimensional view of the particular sample. A 3-dimensional fluorescence image of the particular sample is possible. You have previously in a 3-dimensional structure like a cell or a bacteria, you have put fluorophores connecting at different part of your interest, and then it is lightwise sliced one plane at a time, and then the scattered light from this plane based dissection you are measuring through, say particularly this angle or any other 90-degree angle, the scattered light, and from that you recreate the 3D image. You recreate the 3D image of the slice of the sample and the different amount of scattering at different plane of illumination, and thereby recreate a 3-dimensional image. You have obviously filter sets. Optical filters are used to selectively transmit specific wavelength of fluorescence emitted by the sample. You have beam splitting and steering elements to direct the excitation of the light for the emitted fluorescence, and of course, data acquisition and analysis software. Specialized software is essential for controlling the imaging process and obviously analysis of the data. This provides a very rapid analysis of samples, because instead of doing a point by point scan, you are doing plane by plane scan, right, and immediately getting you slice by slice, you have got a 3D image, a 3D fluorescence image of the sample that you are trying to go through. So, what are the fluorophores used? The present fluorophore is obviously the GFP, green fluorescence protein. There are other yellow fluorescence protein and cyan and red, cyan or cyan, I do not know. Synthetic organic dyes like fluorescence, rhodamine, obviously these days quantum dots. We are using silver nanoparticles, but several other of these type of proteins are used as fluorophores that could be utilized to label to attach with specific areas within the cell, within the bacteria, within the virus, and then slice by slice we are getting information out of it. So, there are different variants of LSM. We have single plane illumination microscopy, lattice sheet microscopy, lattice light sheet microscopy. This is something that I have used, where multiple light sheets are scattered. So, instead of having the illumination from here and detection from here, you have multiple illumination and multiple detection. You illuminate from here, you illuminate from here, you collect the data from here, and you can try to get this information by rotating your detector and your illuminator at different areas, making this as long as this 90-degree angle is almost matched, you can make it in any XYZ direction and thereby get a holographic, get a complete total image of the sample that you are trying to get. So, multi-view light sheet microscopy, ultra microscopy. So, read it at your own leisure that they have been used for taking dynamic processes in 3D in real time, what is happening in a particular bacteria as it is working, the particularly useful for capturing high speed process with minimizing phototoxicity. Scape microscopy, this is imaging fast neuronal dynamics, clearer imaging of thick samples. So, there are several views and all of them utilizes more or less the very same principle, but the amount or the angle of detection and illumination remains more or less 90-degree, but they can use multiple detector and multiple illuminator, just not one detector, one illuminator, multiple detector at different angles and complementary orthogonal multiple illuminators at different angles. Right. So, this is the quite popular one, Zeiss Light Sheet 7. There are obviously several advantages. Light sheet microscopy allows for rapid imaging of samples in 3D, making it well suitable for capturing dynamic processes. By minimizing the out of focus light sheet microscopy provides excellent spatial resolution, can cover relatively large areas of the sample while maintaining high resolution. Light sheet microscopy allows for non-invasive imaging of intact sample, reducing the need for sectioning or destructive preparation method. So, you do not need to destroy the sample, or it's more or less non-invasive, and it's also pretty much, I mean, how do you handle the samples? There are like this holding clamps on which cells are put on a glass, and then you are illuminating it like that, extracting samples, detecting it from like this. You have a small sample bag, or most importantly you can have these cylindrical capillary tubes like I saw before. You put a zebrafishes are usually transparent, or plants or Drosophila fly alive, if you can stick it with something alive but not moving, and then illuminate it from here and get the detection from this particular angle. You can illuminate it from here, you can simultaneously illuminate it from here and get the detection from here and so on and so forth. So, you can have the growth medium of plants, you can have cell glass plates, you can have plastic bags for liquid samples, you can have organism mounting like these kinds of clamps holding the sample. So, all of those things can be utilized using LSM. Fluorescence labeling has to be done in LSM, so that light sheet fluorescence microscope is also used to denote this. LSFM, label targeted structures with fluorescence dyes, antibodies or genetically encoded probes like GFP, and you go for the measurement. So, several different areas can be, applications are there which can be utilized by LSM. LSFM, you have neuronal activity visualization, especially in zebrafish. Zebrafish are optically transparent. You can see the internal organs of a zebrafish from naked eye. You can now fluoresce it with, say, GFP or something like that. So, various areas of a particular organ, especially brain, can be illuminated by this green protein, and then you slice it using the LSM and then measure and then get a 3D image of the brain while the zebrafish is still alive. Yeah. You have connectosome mapping, you have behavioral studies, LSM can capture neural activities where animal perform behaviors or response. This is still challenging. I mean, as the animal is alive, but if it is moving, obviously your focus and everything goes bad, and then you have to redo it. So, like everything else, it has its own challenges associated. Neurodevelopment studies, you take a larva of, say, zebrafish. I know because these are the type of work that I am being doing. You have done something so that the larva has the brain development of the larva at different stages could be used in light sheet measurement, and you can see the 3D image of the brain of the larva, that is the young stage of the zebrafish, at when the age of the larva is 1 day, 2 day, 3 day, 10 days, 1 month, 3 month, 4 month, 6 months, and you can have a real time development of the brain as it passes through different stages of its maturity, right. So, similar other, if you can look into the brain, you can map the brain in a 3D manner, you can look into various diseases that are associated with the brain, neuroinflammation, epilepsy. If you have given some sort of a drug, a pharmacological chemical, and then try to see how it is affecting the brain. If something has attacked the brain, a pathogen has managed to attack the brain, and then a particular set of drugs are being given, how those two are interacting inside the brain at a real time manner. Light sheet microscopy can help you in a very nice way, where a complete 3-dimensional view of the entire thing can be achieved, right. Neurodegenerative diseases, etc., all of those things could be detected. So, in conclusion, LSM's high resolution non-invasive imaging unveils the intricate details of biological processes from cellular dynamics to brain-wide networks, offering unprecedented insights. LSM's long-term imaging capabilities track changes over time, revealing the unfolding narrative of development in neuroscience. LSM opens door to understand neural activity, connectivity, behavior, revolutionizing our grasp of brain. And LSM's fertility spans developmental biology, cancer research, immunology, and beyond, and various disciplines are there to capture the dynamic process. So, these are the concepts that I covered today, and these are the references. Please go through this Zeiss.com's light sheet microscopy. They have several beautiful videos in social media, YouTube, etc., where you can see. They are copyrighted and they didn't decide to give it here, but I will ask you to go and look into light sheet microscopy of mouse brain or zebrafish brain, and you will see the ultra high resolution images of brain and in a 3D format, and what happens to the brain as it performs a particular function, as it goes on developing, as some sort of pathogens have attacked. So, Zeiss have several beautiful images, beautiful videos on this topic. I ask you to search for it. Thank you very much. I will see you in the next class. Thank you.